Today we are going to study related to RNA synthesis and processing. All the very best for your CSIR preparation. So let's start with the RNA synthesis. As we all know that concept of central dogma. Central dogma means how RNA synthesized from DNA and how protein synthesized from RNA. So from DNA to RNA and RNA to protein, that whole process we known as a central dogma. So today we are going to discuss about the transcription. Transcription means how RNA gets synthesized from the DNA molecules. Here in brief you can see, here we can see the double strands of DNA. These double strands of DNA, the DNA get unwinds. Here you can see the bubble. So here we will discuss in brief about what is transcription. Here you can see the DNA strand, double-stranded DNA strand. This double-stranded DNA, first it get unwind, and RNA polymerase enzyme will come. It will synthesize this mRNA. This double-stranded DNA. Next, and little bit brief about the mRNA processing. Like once the mRNA get synthesized, what kind of modification occurs inside the mRNA? If we talk about and that's what we call the post-transcriptional processing of RNA in eukaryotes. So let's start with the transcription process. So transcription means how RNA molecule get synthesized from the DNA molecules. So here you can see the double strand of DNA. First of all, the melting happens. That means the unwinding of DNA happens. So in this DNA strand, the RNA polymerase will come and it will start synthesizing mRNA molecule from it. Next is, once the mRNA gets synthesized, next process happens is its processing. That's what we call the post-transcriptional processing of RNA in eukaryotes. So majority, three kind of modification happens. One is, see, eukaryotic mRNA contain intron as well as exon. Intron we know that they are non-coding sequence. They do not code for any kind of genes, while exon are the DNA sequences which code for a gene. The genes will convert it into the protein subsequently. So first thing is the splicing. In splicing method, what happens? This non-coding region of intron will get removed from the mRNA sequence. Then the second thing happens. So this is intron. This is also intron. So these introns will get removed. The next thing is poly-A tail attachment. So in mRNA sequence, if you can see here, at the 3' end, poly-A tail will happen, and at the 5' end here. That's what we call the 5' cap, because here in eukaryotic mRNA it has residue of 7-methylguanosine linked at 5' terminal. 7-methylguanosine. So these three kind of modification happen during the mRNA processing. Next, so let's go through one by one each quiz. I will give you a time to think on it, and you have to answer it after 15 seconds. Also, we will discuss about each question once you answer it. Okay. So first of all, among the following, which statement is correct regarding the promoter of the gene in eukaryotes? They have give you 4 sentences related to promoter in eukaryotes. You have to find out the correct one. Which sentence is correct? So A, it is always located upstream of transcription start site. B, it is always located downstream of the translation start site. C, different promoter elements are recognized by different RNA polymerase. D, promoters are recognized due to their different secondary structures. Your time starts now. You have to think about correct sentence. Which one is appropriate for eukaryotic promoter? Your answer is C. Different promoter elements are recognized by different RNA polymerase. So that means the sentence A, B and D are not correct. Means it is not necessary that it will always be in upstream. It is also not necessary that it will be in downstream of translation site. And D is also not correct. So the right answer is C. Next. In bacterial RNA synthesis, which of the following is the function of factor Rho? So here they have asked about prokaryotic RNA synthesis. RNA synthesis means transcription process, and rather than eukaryotes, here we have to discuss about prokaryotes. These are the four options. A. Allow proper initiation of transcription. B. Eliminate the binding of RNA polymerase to the promoter. C. Participate in the proper termination of transcription. D. Increase the rate of RNA synthesis. Your time starts now. Here you have to think about the function of Rho factor. You should have clear-cut idea what does it do, where it function, what is the exact function of Rho factor in bacterial transcription. So the answer is C. It participates in the proper termination of transcription. So the main function of Rho factor is in the termination process. I would like to tell you something about the Rho factor in case of bacterial, or we can say prokaryote transcription termination. See, when we talk about the prokaryote termination, it's always of two kinds. One is Rho dependent and one is Rho independent. And as they have asked about its function, so if we talk about Rho dependent termination, what does it do? This Rho dependent termination uses CA rich sequence. See, remember it uses CA rich sequences like this. Such sequences are getting used by this Rho protein, and this sequence is we call Rho utilization or RUT element. That is RUT, RUT element. Okay. So this Rho protein associated with these sequences, and it hold the RNA polymerase there, and it allow the mRNA transcript to release from these DNA sequences, and it causes the termination of the mRNA synthesis, or we can say termination of transcription process in case of prokaryote. So that is all about the Rho factor. Once again repeat, in eukaryote there are two kinds of termination in transcription. One is Rho independent and second is Rho dependent. In case of Rho dependent termination of transcription, it is related to Rho element. Rho element is CA enriched, and that's why we call it as a RUT, means Rho utilization elements. Now next question. Which of the following is or are true about the eukaryotic transcription? Earlier we have seen about the prokaryotic transcription. Now they have asked about eukaryotic transcription. So first is, RNA polymerase needs general transcription factor to initiate transcription. 2. Tissue specific transcription factors bind to data box. 3. General transcription factors bind to long range regulatory element. 4. CTD of RNA polymerase II plays important role in transcription initiation. Read carefully all four options and give the answer. Your time starts now. Once again I am repeating, they have asked about the true sentence about eukaryotic transcription. Choose wisely your option. So here is the answer, which is sentence AN4, means the A is correct. RNA polymerase needs central transcription factor to initiate transcription. And the fourth one, CTD of RNA polymerase 2 play important role in transcription initiation. As we know that in case of eukaryote, the RNA pol 2 play a very important role in transcription process. And if we talk about the first option, which is about the transcription factor, because we know that transcription factor are required to initiate the transcription process. So in case of eukaryote, if they have asked about the first of all which transcription factor comes and bind to it, so that is transcription factor 2A. So RNA pol 2, for example, it is a DNA. So, and this is Tata binding site, TA. So this RNA pol 2 enzyme will first come to this site and it will bind here. This binding of this pol 2 and Tata binding site, this RNA pol 2 will come to Tata binding site by the help of Tata binding protein. Tata binding protein is TBP. So this Tata binding protein will recognize this Tata box. It will help to recognize it. So we know that the D is the answer. That means option 1 and 4 is correct. So now let's understand what happens actually during the eukaryotic transcription happen. This is the DNA. In this double-stranded DNA, there is a Tata binding site. So first of all, the RNA polymerase 2 enzyme is required to initiate the transcription process. It will not be able to find this Tata site. So it requires a protein, and that protein what we called is Tata binding protein. This TBP, that protein will help this pol 2 to recognize this Tata binding site. So after finding this Tata binding site, the first of all the transcription which is going to be used is transcription factor A, TI2A. That will first come and it will help RNA polymerase 2 to initiate the transcription. Now next. In eukaryotic transcription by RNA polymerase 2, formation of a pre-initiation complex. Here are your options. Begins with the binding of TBP to Tata box of promoter. B. Involves the ordered addition of several transcription factors and the RNA polymerase. C. Allow an ATP dependent opening of two-strand DNA. D. All of the above are correct. Your time starts now. See, recently we have studied about it. So here you can see about eukaryotic transcription, RNA pol 2 related things. Read carefully each and everything. We have just discussed about TBP, Tata box, each and everything. So D. That means all the sentences are correct. So let's go through one by one. In case of eukaryotic transcription, we have just discussed, like it starts with the binding of TBP, which is Tata binding protein to the Tata box. Second is, it involved in order addition of several transcription factors. So which is also correct, because we have just seen first the transcription factor A will come, then transcription factor B will come, then transcription factor E will come, then transcription factor F will come. So like this, many transcription factor will come and it will help to complete this transcription process. Also, it allows ATP dependent opening of two DNA strands, because we know that DNA is double stranded. So RNA pole binding of RNA polymerase enzyme to this DNA, it requires the unwinding of this double stranded DNA, but it should get converted to single stranded DNA. Then only RNA pole will come and it will get attached to it.